What we're going to talk about, I'm joined by Charlotte Pawlyn, who will introduce herself properly in a moment, I'm sure, but who's a consultant at the ICR at the Royal Marsden and a group leader at the ICR. My name is Parrish. I'm one of the haematologists in Leeds and also based at the CTRU at the University of Leeds. And what we're going to talk about today is a bit about our personal experiences, career experiences so far of being early career investigators and our involvement in clinical trials research and how we've become involved in clinical trials research and are interested in it. And really the aim of today's session is to give a bit of an idea about portals and routes that you might pursue if this is a kind of area that you're interested in. And so what we're planning to do is to give a couple of fairly brief presentations about what's happened in our careers so far and how we've become involved personally. And then I would quite like to open this out into a discussion. If anybody has questions about this or wants to know a bit more about different aspects of this or how they might get involved. I know everybody is by now extremely familiar with online meetings and webinars and so on and so forth, but if anybody is not muted themselves, can you please do so during the talks? And then what we'll do is at the end of these presentations, we will turn the slides off and let everyone to put their cameras on, and happy to sort of have a discussion at that point. And if there are questions that come up as we go along, please feel free to put them in the chat and I'll try and collect them, and we can field those either at the end of the talks or interrupting, depending on what's most appropriate. Okay, thank you. So without further ado, I'd like to introduce Charlotte and I'll let her kick off the session.
Thanks, Chris, and hi everybody. Thank you for joining us on this very sunny Thursday afternoon. So my name is Charlotte Pawlyn. I'm a haematology consultant at the Royal Marsden Hospital in London, and I am a group leader and CRUK clinician scientist at the Institute of Cancer Research, which is where my laboratory research is based. So I've sort of taken the approach of trying to think about what might I have wanted to know when I first started thinking about how I might get involved in clinical trials research. And what are the things that I think that people might want to be aware of, or the kind of routes in, or the things that might help you progress along that pathway. And when I was thinking about it, I think that there are three kind of key elements of what has been helpful to me in terms of getting involved in clinical trials research. And those are kind of formal training, so ways in which you can acquire some of the skills and knowledge that you need to be involved. Then experience, so getting some of that initial experience and then building on previous experience to try and develop as a clinical trials researcher. And then the third most important thing for me along the way has been mentorship and support from people that I've met whilst working on clinical trials, who've then pointed me in the direction of more training or more experience or other ways in which I could get involved. And so for me, these things are still ongoing. I'm still training, I'm still gaining experience, and I'm still heavily reliant on mentorship and support from other people, for which I'm very grateful. And I can see that for the rest of my career, these things will all intertwine and support me to hopefully become more experienced, more competent, more developed in what I do. So if we take each of these in turn, I'm going to start with experience in terms of how you might think about getting first involved in clinical trials. So apologies for the non-medics in the audience. I guess this is a heavily kind of medical training orientated talk, because that is mine and Chris's experience. But in terms of how, as a trainee or a consultant, you get involved in clinical trials, there are, I guess, two kinds of trials that we're often exposed to. So both commercial studies run by pharmaceutical companies predominantly, and then academic studies truly designed and led by academics, clinicians, and clinical trials units in the UK. So one of the routes in would be to be a sub-investigator on clinical trials as either a registrar or as an early career consultant, either within a commercial study or within an academic study. And then as you gain more experience or become a consultant, it's more likely you might be able to be a principal investigator at a local site, and subsequently, hopefully in the future, a chief investigator for a commercial study within the country or the overall trial. From an academic study perspective, there are kind of other ways, I guess, to get involved. So one of these that I just wanted to highlight is that the NCRI, who manage the subgroups that just help design clinical trials in the UK, of which the HEMONC subgroup is one, run a trainee experience programme. And this is something that I took part in as a registrar. So you are allied to the group for a few years, and that exposes you to how academic clinical trials are designed and set up and then run. But again, there are sub-investigator positions within lots of hospitals that run clinical trials. There's the UK-Myeloma Research Alliance, which we'll talk about as we go along, but a group of clinicians across the country interested in setting up and designing academic clinical trials. There are potential possibilities of getting involved in trial management groups through that membership, and then potentially going on to design clinical trials, protocol writing, or taking on more senior roles within trials through that. Aside from experience, I think there are some kind of key components of training that are available to gain out there that help us to develop the skills that we need to do this. So obviously the very basic is good clinical practice training, which is obviously essential before you work on any aspect of clinical trials. But then in terms of kind of more extensive training, there are statistics courses, data analysis courses, research methodology courses, all kinds of things that are available more and more online to be able to get some of the skills that are going to help with progressing along this way. I guess one of the things I also took part in early in my training was local site initiation visits or investigator meetings. So as new studies are opening, really engaging with the process of understanding how that study is being set up in your centre, so that you then understand the processes that if you were to go on and design a clinical trial, your trial would need to address and enable the local centres to follow. And then I just wanted to highlight a couple more formal training aspects that are out there to help in terms of clinical trial design, protocol development, protocol writing. One of these is the Methods in Clinical Cancer Research, which is run by ESMO and AACR. This is where I first met Sarah Brown, who set up these webinars, and this is my group from that meeting, where we spent a very intense few days writing an entire protocol and learning how to do that with the help of experts like statisticians like Sarah, but also clinical trialists from around the world. There's another equivalent run by the American Society of Haematology and European Haematology Association called the Clinical Research Training Institute, which is a similar concept in terms of a kind of intensive few day long trying to write a protocol and take it forward kind of course. In terms of mentorship, I'm incredibly grateful to all of the people who've helped me get enthusiastic about clinical trials along the way. And this is a list of, when I started writing this list, it was shorter, and then it kind of kept getting longer and longer. And so I first got into clinical trials when I started my registrar training, and was really supported by the people within the team within my hospital. So the data managers, clinical trial administrators, clinical trial coordinators and research nurses, who will kind of help me understand what my new role was going to involve and how to go about doing that. And then clearly I've had a lot of support from other clinicians, so sub-investigators, principal investigators and chief investigators of trials along the way. But I've also learned an incredible amount from statisticians and the clinical trials units, who are all experts in all aspects of this. And I've learned an incredible amount from them about how to work in this area. But most of all, I think I can think of key patients along my career who've, especially when I was starting, probably taught me more about the process of clinical trials than I knew at the time. So we had some patients who have been in several trials, and the nature of myeloma clinical trials is such that that is often the case, and learning from their experiences of how things were done well or not done well potentially previously has also been really valuable to me. So I think all three of those aspects are really important if you're kind of thinking about how to get involved. And the next question that I thought about in terms of how to address this question was, well, when should you try and do this? And this, for the non-medics in the audience, is a kind of probably now slightly out of date outline of haematology training in the UK. So leaving university, doing some foundation year and core medical training, and then going on to do specialty training. So for example, for myself in haematology, before completing training and going on to be a consultant. And we obviously have exams to take along the way and numerous other things that need to get done. So when is the right time to start doing this? So I guess the answer to that is different for different people. I started myself at the beginning of my specialty training. So when I started training in haematology, I became involved in clinical trials from that point. But I also know of consultants and even more senior consultants who start to get involved later in their career and really get involved at that point. So there's never a wrong time or a right time. If it's something that you think would you'd be able to add value to and be enthusiastic about and want to get involved with, then now is the right time. From my experience, just to give an example of how some of this can work along the way. So I was a NIHR clinical trainee. So I started my haematology training with something called an academic clinical fellowship. And that gave me some 25% of my time in my first years of training that was dedicated to research. And I spent some of that time doing clinical trials research and some of that doing laboratory research. And my laboratory research led to me going in to do, my graphic hasn't appeared, but going in to do a PhD, which is what this box would say. And part of my PhD was learning how to perform experiments in a laboratory. Part of that involved the analysis of clinical samples from one of the large UK clinical trials. And ended up in me writing a thesis about epigenetic control of multiple myeloma and how we could target that. That led on to me continuing the rest of my training as an NIHR clinical lecturer, which by that point gives you 50% of your time, and in research developing either laboratory or non-laboratory research skills along the way. And I continued that, and then was fortunate shortly after I had my consultant appointment to continue my the laboratory aspect of my research career. I now have my own lab in the Institute of Cancer Research. We recently moved labs, which is what this picture denotes. And the goal of my lab research is really to take ideas and discoveries from laboratory-based research projects and translate those into approaches that we could then study in clinical trials and translate that into a therapeutic benefit for patients. And so you'll note there's not a lot of clinical trials going along here. So I just wanted to highlight how that kind of integrated with the training and the laboratory research that I was doing. So as I mentioned, when I started at ST3, so when I first started haematology training, I became involved as a sub-investigator of both commercial and academic studies within my centre. During my PhD, I became much more involved in several of the academic studies led by my PhD supervisor, so myeloma 11, MUK4 and MUK7, and I became involved doing response reviews for patients, contributing to protocol amendments and participating in pharmacovigilance teams, so looking at adverse event reporting within the trial, as well as contributing to analysis of data from the trials for publication. But the main example I wanted to give is of the fitness trial, which is myeloma 14, which is something that I've been involved in for many years now, and I think illustrates well how if you think you want to be involved in clinical trials, but take that further to try and start designing your own trial and setting up a trial, then this is a good example of that approach. I should mention that the chief investigators of the trial, of the fitness trial, are Graham Jackson and Gordon Cook, who've provided me with lots of mentorship and support, but I've been involved along the way with the trial design and setup. That's the picture. So the fitness trial myeloma 14 is recently opened, so opened in August of 2020, and the trial aims to study the population of patients who are not fit for autologous stem cell transplant, so older, less fit patients, and study whether or not we could improve their outcomes by adopting an adaptive frailty score adjusted dosing strategy. So rather than patients all being given standard doses of drugs, we are categorizing patients based on their level of fitness and delivering dose modified induction therapy to see whether that will enable patients to remain on therapy with fewer adverse events for longer and hopefully improve their outcomes. And there's a second randomization after 12 cycles of induction to the combination of lenalidomide and ixazomib or lenalidomide alone. So the trial is really based on some data that we studied from the Myeloma 11 trial, which was the precursor, or the preceding study, if you like. And we looked at data on transplant ineligible patients within that trial in 2013, so 8 years ago now. And we explored some of the toxicities dependent on age group within that trial. And it appeared that patients in the older age group were suffering with more toxicity, and we thought about ways that we could try and address that to try and enable them to stay on therapy for longer. So after a few years, we'd formulated some ideas, had some discussions with pharmaceutical companies and discussions within the NCRI subgroup, the UK-MRA-MUK Research Alliance. About that time, a paper was published looking at ways in which we could classify frailty amongst patients, which gave us a way to do this in a less age-based and more patient individualistic approach. So we applied for funding from CI UK, first in draft and then in full, and then spent several years developing the protocol, patient information sheets, informed consent forms, designing translational research studies, gaining all of the reviews and approvals that you need for trials, and then finally opening the study at the end of last year. Along the way, we did some more data analysis from the Myeloma 11 trial to try and kind of firm up our hypothesis and really support the underpinning of the Myeloma 14 fitness trial. So I guess what's important is that this is quite a long time. It's not a process that happens terribly speedily. Perhaps we might wish that that could be faster, and we've seen examples from the vaccine trials of how some of these things are hopefully changing over time. But a lot of this was out with our control, and the important news is that the trial is now opening and recruiting patients around the country really successfully. So I guess just to finish, if you have the question, you want to get involved in clinical trials research, then I would use our approach to patients, which is it's okay to ask people. Please ask us, please ask other members of the UK Myeloma Research Alliance. Please email me if you have any questions, and we'd be more than happy to help along the way. Thank you very much.
Thanks, Charlotte. That's a fantastic overview. Are there any questions for Charlotte right now? I don't see any in the chat at the minute. So what I think we maybe might do is if I press on and give my talk, and then we'll open things up for some questions at the end of that. So I'll just share my screen. Okay, so what we thought would be helpful is to show you that perhaps there are certainly some similarities in terms of the paths that Charlotte and I have taken, but actually also some differences too in terms of what we have done along the way and what we do now, and really to point out really some of the different ways in which you can get involved in clinical trials and the different avenues that are open depending on what you're doing at the moment. So in contrast to Charlotte, my job's sort of the other way around. So I am a NHS appointed consultant, and I have now a little bit of research time at the University of Leeds, but I'm predominantly an NHS appointment. And so I'll talk you through a little bit how what's happened to me in terms of my career and the things that I think have been useful. Obviously, being an early career investigator myself, but being, the things that have been useful so far in terms of trying to get into clinical research and avenues that I think might be helpful for people that are interested in this. So having completely, so I thought it'd be useful to lift Charlotte's slide and just show you a little bit about how I've sort of approached this. So I've had a slightly different route, I suppose. So in terms of my training up to being a haematology registrar, I wasn't in an academic training role. I went through a standard deanery training post, and indeed went into a standard deanery haematology training post. And I started my haematology jobs. So for those that aren't medics, ST3 of these now go up to 7, and things keep getting renumbered and constantly changed about once a year, just to make sure that nobody really knows what anything is called or what's going on. But essentially, this is haematology registrar training. So and I went in and I started in a District General Hospital in Yorkshire, doing, learning about what blood films are and how to deal with people who are neutropenic, and sort of did that for about 18 months. And then at that point, what I did is I rotated into Jimmy's, which is St James's Hospital, which is the big teaching hospital in Yorkshire. And I started off doing a bone marrow transplant job, actually. And the things that immediately happened, because I was in a big teaching hospital, really, was that I started to realize that we have a huge number of patients that are on clinical trials here, do various different trials in different areas of hematology. And which is very interesting, because you come across a lot of things which are new and you've not encountered before, and not happening in other hospitals. And so I became quite interested in this and tried to find out a bit more about the trials and what was going on. And of course, as Charlotte mentioned, the first thing that you need really to be involved in trials is to do good clinical trials practice training, which teaches you about some of the basics of how trials work, and the sort of surrounding legislation and the logistics of running trials. And because I'd done that and sort of taken an interest and was interested in the trials, and because we have a very active trials team, I was able to get involved on some of those trials as a sub-investigator, to attend some investigator meetings. And I think that once you start to get involved in the trials, you start to become interested in the way that they're run, and it starts to generate ideas yourself of things that you might do. And one of the first things I think that emerges is once you start to sort of get in the way of people that are doing these things and make yourself known, people will start giving you jobs to do. So the first thing that happened really was that it was suggested to me that, I was quite interested in this group of patients. Well, why don't you have a look at the outcomes of this this group of patients and see what's happening in these elderly patients that we were giving bortezomib at the time? And so I looked at a local case series, wrote that up. And this is not a high impact Nature publication. This was a letter to BJ Heme, but it was a useful sort of first step to start to get really some interest in it, and to generate a bit of traction, and to develop a disease specific interest, I suppose. And so because I've been involved in that, I started to be a bit more interested in some of the other myeloma patients and the treatments that we were giving. And I was doing a transplant job, and then I went on to a myeloma job. So I was doing a lot of autograft work, and started to look at some of the autograft patients that we had in our department. And particularly wondering about what happens to these patients who don't actually respond very well to transplants. So what I did then was I got in touch with the BSBMT network, the registry, and started to try to get access to our national data to have a look at actually what happens with these patients who have suboptimal responses to induction therapy, what happens when you transplant them. And what you'll find if you do things like this is that generally people are absolutely delighted if you've taken an interest in an area and you've got a bit of time and resource and enthusiasm to throw at a topic like this. So they were really helpful, provided you with lots of data, lots of statistical support to write things up. And then this was a poster which I presented at ASH. So I'd started to sort of think that I might be quite interested in myeloma at this point. And the next thing that I would say, and I agree with everything that Charlotte said, one of the things that's important in trying to develop an interest in trials is mentorship. And there are mentors that are in all sorts of areas of what we do, but I think it can be really useful to tap into the experience of people who really know what's going on, and to if you show that you're enthusiastic and you're interested, people will be only too happy to make use of you as a resource, and to build you into things and to involve you. So I think that's really a kind of a key message from this. I think that's important. And the other thing is, again, I agree that teams are really, really important here. And one of the things that you learn as soon as you start to get involved in trials is that it requires huge numbers of people to run effective trials. And what you need to do is to start to begin to build networks with those people and get to know who's doing what, and involve yourself in different aspects of it. So at this point, I lost my cursor. So at this point, I decided I'd done my first 3 years, and I'd done my exams. I was fortunate enough to get those. So I thought this was quite a good time to do my to take some time out of program. And I decided to start doing a PhD at that point. And so I applied for a PhD and took some time out working with Gordon Cook. And I started to look at T cells, and I started to look at a group of T cells that are, in retrospect, a very rare and very difficult to characterize group of T cells, which I ever since had a love-hate relationship with, but started to get interested in, you know, the sort of T cell biology that is important in myeloma. And so I started to do bits and pieces of work looking at immune reconstitution and what's the role of the immune system in fighting cancer. And because of the groups that I was working with, I was working alongside a group that were looking at using an oncolytic virus, called, in this case, a reovirus, which they were using as a sort of potential treatment for solid tumors. And so, again, trying to sort of build networks with other people that are around, the group there were very interested in expanding that into hematological oncology. So I started to look at this virus as a potential treatment for hematological malignancy, including myeloma. And that led to a sort of a larger bit of work, and I decided that what would be useful is to extend my out of program experience. And I spent a bit of time at the Mayo Clinic with a collaborator there, sort of building up this evidence base for reovirus as a treatment for myeloma. But whilst I was doing all this, because of the interest really that I had in autograft, so I was quite keen to maintain involvement in that. And so what I started to do was to get involved in clinical trials. And the route really that I took here was that I was involved in doing the external data verification. So essentially this meant looking through the response parameters for patients on trial and checking the central and local lab results to make sure that the trial reported outcomes were correct. And that's not a particularly glamorous job. It's quite a lot of work actually, and it's actually not as easy as it sounds, I would say, either. But it was quite useful because I got to know the team that were working on the trial, and I got to be involved in the study, and to get to know a bit more about it and to build the networks. So the one thing, I suppose this is not really supposed to be a potted CV, more to try to point to the things that I think that were helpful here. And one of the things that's really important is to build networks. So to start to be in touch with the people that are running trials and tell them that you're interested and that you have you know, time and enthusiasm to offer. And I think if you do that, then people will involve you. So this reovirus work was quite promising. We'd done quite a lot of in vitro work that showed that essentially this virus and this combination could kill myeloma cells, and it worked quite well with immunomodulatory drugs, and we've done some in vivo work to show that. So to cut a long story short, this really looked like quite promising experimental therapy for myeloma. And so as a result of that, we approached Myeloma UK Early Phase Trials Network, so that was the precursor really to the CARP program, and designed this study, which was a MUK trial, the MUK-11 study. And the idea here was that we were going to use patients who had immunomodulatory therapy with either lenalidomide or pomalidomide, who were having serological progression, but not really clinical progression. So people that were starting to get to the point of needing to change therapies. And rather than swapping them completely to the next line of therapy, to try stopping their steroids and instead augmenting their IMiD therapy by adding in this reovirus on the basis of the preclinical data. And so we built this study up, and I have to say that I learned an enormous amount doing this, you know, writing the protocol. I had not been on protocol writing courses or clinical trial designs courses, and actually that is something that I would strongly recommend that you do. I think that anyone that's interested will find those very, very useful courses that will teach you a lot. But I did, on the other hand, have an enormous amount of help from the team over at the CTRU and others to sort of put this protocol together. Now, unfortunately, this story did not end particularly well because MUK-11 was plagued with a few problems. Namely, the virus turned out to be quite difficult to deliver in terms of a clinical agent, and most of the sites that had initially thought they'd be interested in the trial were unable to open, so we weren't able to recruit to the study. So it was I suppose, a sobering lesson that clinical trials can be quite difficult to run. They are time consuming, but actually, also that is exactly the reason why you need these networks of people to help you, and experts who can guide you through these areas. And if you ask for help, there is lots of help available. And I suppose it's also an important lesson that the first idea might not be the best idea, and these things, as Charlotte showed you with the design of fitness, these things are iterative and do take time. And I think perseverance is an important attribute if you want to be involved in this sort of work. So I did that. I finished my out of program experience and I went back into training and I completed training. And then essentially there's a question about how do you want your career to progress from that point? And there were really options of, do I continue trying to do laboratory work and continue for example, the work that I'd done on immune biology, or did I look at taking an NHS consultant role? And I think that the choices here are going to be different for different people. There are pros and cons to both of those, and that this talk isn't really supposed to be about how you should base your career, more about saying that actually it doesn't really matter which of these routes you take and how your career pans out. There are opportunities for people to be involved in clinical trials research, irrespective really of which of these routes you take and how much of your time you have to dedicate to it. So what I did is I took an NHS consultant post at St James's, and this is where I work now. So the question is, having done that about 5 years ago, what are the ways in which as a NHS service role, so as an NHS consultant, what are the ways in which I'm now continuing to be involved in clinical trials? So the first thing is working as a local investigator, and we've talked a bit about this already. And we're lucky in St James's in that we've historically had a very large and productive trial portfolio. I have to say that at the moment, as with many other centres, this is a difficult time to run trials because of everything that's happened with coronavirus, and we, like everybody, are struggling at the moment to get open all of the studies that we'd like to do. But nonetheless, I think those problems will pass, and I think it's one of the things that's really rewarding about my job is being involved in the late and indeed the early phase trials. And the early phase trials are often as local collaborations with the university or with smaller companies. We talked a bit already about GCP, and we talked also about site initiation visits and investigator meetings. I think those are all important ways to get involved in the trials and to work out what's going on. And lots of us have the opportunity for local research projects and pilots. And one thing I would say is that those that quite often give rise to ideas that can then spin out into trials and can be shared with other people. So those are useful, I think. But the other thing that I spend quite a lot of time doing now is that there are lots of people in the UK that are running a great number of extremely impactful studies in myeloma. And we have a big, very inclusive research collaborative, the UKMRA-MUK Research Alliance, that I would encourage people who are interested to join this and to find out about the trials that are running, and to attend UK trials meetings, to find out about what the registries are doing, what the special interest groups are doing, to be building up networks of interested individuals and involving yourself in those. And I think, running trials, anybody who runs trials will tell you this is hard work and there's a lot of work involved, and people are needed on these trial management groups to take on different aspects of that and to contribute to the management of studies. So if you are interested, those are certainly avenues that you can take to being involved in the studies that other people are already running, and there will be various ways that you can contribute to those, developing the studies and adding value to them. And I suppose the holy grail really is that is coming up with ideas yourself for how you're going to run studies. And I think I wouldn't want you to think that unless you've got an idea for a fantastic clinical trial, go home now. I think that if you get involved in things, then you will appreciate, you'll come to realize some of the issues that are going on, and ideas will follow to a certain extent. So, but I think when you have ideas, we have an inclusive group of people who are really interested in sort of discussing things. And one of the main things that we want to get across from these seminar series really is this webinar series is about how you go about bringing these ideas together and proposing them. And if you look back through the archive of the previous webinars, you'll see an earlier webinar with a really clear where Sarah's gone through a really clear explanation of exactly what the process is of applying and submitting expression of interest to CARP. And the take-home message really is that there's a lot of help here available for people who've got ideas for studies who want to run things, in terms of you don't need to know everything about running a trial. There's lots of additional resource that can be brought to help you if it's felt to be an idea which is potentially very interesting. So I suppose the things I would say are important. I think enthusiasm and persistence, perseverance are important if you want to work in this area. It isn't necessarily a quick win. I think there is, and help and mentorship are important, but I think that you don't necessarily have to be at a hospital where you have a a leading myeloma expert working there who can guide you. I think that actually, if you look across the UK, you're going to find that it's a very inclusive group of individuals who will be very welcoming to anyone that has ideas and wants to contribute. And certainly, I know either of us would be happy to be contacted and to sort of point you in the right direction if there are ideas and contributions, and if there's way that we can help. So take the opportunity to get involved. I think if you have ideas, that's absolutely fantastic, but actually also people that are enthusiastic and want to get involved in trials and don't necessarily have the idea for the next big trial, still welcome, and we'd like to hear from you. So that's all I was going to say for now. What we thought might be helpful is that there may be people who've got questions about any of this or want to discuss anything that we've said in any way. So if there are any, if anyone has any questions or things that they'd like to talk about, now is a good time. The thing about these webinars is everyone's with their camera off. We're hoping that they're all still awake, but.
If not, please feel free to email or contact either of us afterwards.
Perhaps I'll maybe start, Charlotte, in terms of sort of the courses and things that you've done as you've been going through. Are there particular things that you point to that you'd say had been really useful? Things that kind of guided you in how to approach trials?
I think the clinical trials writing course that I went on, as you said, I mean, I think it's such a complex process if you've never kind of approached it before. It was a very intense experience, and trying to write a protocol in a week is not easily undertaken, but it was incredibly useful in terms of understanding the kind of approaches, the statistics, every aspect of it. So, I have to be honest, I've not put all of it into practice, but I think having an understanding of it is really important to then understand how other people are contributing to a protocol that you're maybe working on together. So from that point of view, that was probably the most valuable thing I've done in terms of kind of formal training.
So I think that can be quite a daunting thing, can't it, when you have an idea, but actually the kind of steps between the idea and running a clinical trial are a bit bewildering if you've not done that before. So. Okay.
I'll ask you a question then. Has your experiences of MUK-11 put you off wanting to do this again?
So I think MUK-11 was quite it's a disappointing experience when you put a lot of effort into trying to run a clinical trial and it doesn't happen. But actually that's more common than probably many of us realize, and lots of clinical trials will fail before they get to the point of recruiting patients, or even worse still afterwards. And I think that you have, I can't really pretend that it wasn't disappointing, but on the other hand, I think that you do learn a lot about how trials work, and it's important to just sort of take that learning and employ it with the next time round. And I think we learned quite a lot about how to run and how not to run early phase trials from that. So okay, well, I think if there aren't any questions, shall we wrap things up there? And what I would say, I think Tyrion might have a closeout slide for us. There will be another webinar in this series. The date is fixed. The exact content, I think, of the webinar is not yet, so that's to be announced, and there will be some information sent around about that in due course. And you can, of course, you can see this and the previous webinars on the YouTube channel, and you can also see that on the web link to from the website. So if you want to look back, and I would encourage anyone that has ideas to look back through some of the previous webinars that will give you information about how you might go about sort of approaching people and implementing it. So thank you all for joining us, and we'll let you go into the sun now.